I'd like to again welcome everybody to now day 2 of this fabulous symposium. I myself am Rob Milechka. I am the chair of the chemistry department, and I really want to welcome everybody, especially those of you who have come from very far away. Before I turn the mic over to the provost for the official welcome, I want to recognize some special guests in particular, and that is Professor Rosenberg's former students. What we're celebrating here today was really a world-changing event, and that is, of course, the FDA approval of Cisplatin. But it's impacted, obviously, millions of people, patients around the world, and of course it's impacted those of us here at Michigan State University, and that certainly started with that initial group of students from the Rosenberg labs. And so what I'd like is that we have 15 former students of the Rosenberg labs here with us today, and I'd appreciate it if you would all stand up, and so if we could give you all a round of appreciation. And with that, I'd like to turn the mic over to June Youat, Provost, Michigan State University.
Well, thank you, Rob. It really is my privilege to be able to welcome all of you and thank those of you who have traveled a very, very long distance to be part of this. At Michigan State University, those of you who've come back home, you know that we pretty often enjoy celebrations of key marker events in the life of the institution, in the work that our scientists, researchers, scholars do, and in all kinds of victories. But this is a particularly special one, as Rob has suggested. The impact of what we celebrate today, the approval of Cisplatin by the FDA 40 years ago, really has changed the landscape of this country, of the world, and of individual lives, and I know that's a piece of what you'll be talking about today. I want to acknowledge, and I'm not sure she's in the room yet, but we have a very special guest, and I hope you'll have a chance to meet Barnett Rosenberg's daughter, Tina. Is Tina? Ah, there she is. Welcome. We're so happy you're here. Besides the fame at which she walks in the room as his daughter, I think many of you know that she's a very accomplished journalism and author who's appeared in the Republic, the New Yorker, the Washington Post, a fellow at the World Policy Institute, and an editorial writer for the New York Times. And she's also the author of 3 books. One of them, The Haunted Land, Facing Europe's Ghosts After Communism, won the Pulitzer Prize for General Nonfiction and the National Book Award. So again, welcome. It's really an honor to have you here. So today's symposium, as you know, will showcase the worldwide importance of both cisplatin and carboplatin, effective drugs whose anti-tumor activity was first discovered in the laboratory of Professor Barnett Rosenberg here at MSU in the mid to late 60s. Then subsequently approved by the FDA 40 years ago, really revolutionizing the treatment of certain cancers, especially testicular, bladder, lung, stomach cancers, and saving countless lives. The presentations you'll hear today are really a who's who of scientists and experts who participated in these agents from the 60s to the 80s, including both US and international scientists. And of course, there'll also be presentations from MSU faculty members from today who will share some of their latest cancer research advances in precision nanomedicine, in medicinal chemistry, and metabolic regulation of this complex disease. Beyond the lives that were saved by cisplatin, we also have been so appreciative of the benefits of that at Michigan State University, because the proceeds from that have really funded decades now of scientists in their work and also in the transfers of their medical technologies. And you'll hear a little bit more about that today from David Washburn after lunch, as he talks about the foundation of the MSU Foundation, which was created actually to handle some of the royalties from the FDA approval of cisplatin. So again, thank you for being here. It looks like a pretty remarkable day. I know some of that time will be spent talking about what has been, the past, celebrating that, reminiscing about that, and much of today will be about the future and how what has been done really serves as a prelude to all that will be done in this important area. Thank you again for being at Michigan State. It really is an honor and a pleasure to welcome all of you.
Thank you, June. So as we all know, this is a scientific symposium, and an important part of any scientific meeting is the opportunity for discussion. And so we'll be operating similar to Gordon conferences for those of you who have been at those events. And we will have discussion leaders that will help prompt the discussions. There will be Sarah and Mary will have microphones. So if you have questions after the talk, please raise your hand. They'll bring the microphone to you. To help facilitate those discussions, we will have discussion leaders. As I think most people know, Professor Rosenberg was a biophysicist, and I thought it appropriate to have our first discussion speaker, our discussion leader, I should say, be a biophysicist. And so with that, I'm going to turn the mic over to Professor Matt Comstock.
Right, so I'm a new generation biophysicist, which means these days we make fancy microscopes with lasers and grab onto the little molecules that interact with your DNA and watch them walk around and keep cancer from happening to you sometimes. I'm over in the physics department, and I'd like to introduce our first speaker today, Jim Hoeschele, who was a former student of Arnie Rosenberg and is currently an adjunct professor in chemistry at Eastern Michigan University. He's going to talk to us about the discovery of the anti-tumor activity of cisplatin. Thanks.
Thank you, Matt. Thank you, Matt, and thanks to all of you for coming today to witness this 40th anniversary celebration of the FDA. To witness this celebration of the FDA approval of cisplatin. It is especially, I'd like to, again, as Provost Schuott said, to thank all former students of Dr. Rosenberg, 15 of whom are here attending this talk. The title of my talk is Serendipity and a Prepared Mind, the Discovery of Cisplatin. The plan is to first pay tribute to Dr. Rosenberg and Loretta Van Camp, and to acknowledge the research contributions of the Rosenberg students who actually participated in the discovery and or development of cisplatin and carboplatin, as it turns out. And then finally discuss the broader impact of this momentous discovery. Professor Rosenberg, who was indeed a brilliant and creative scientist who had a long and distinguished career. His curiosity, imagination, and persistence led to discovery and development of cisplatin, a globally important anti-tumor drug, often referred to today as the most important anti-cancer drug ever discovered. Professor Rosenberg was educated in New York. He obtained a BS in physics at Brooklyn College, an MS and PhD in physics at NYU. He joined the department at MSU in 1961, co-founded the biophysics department actually with Leroy Augenstein. He was indeed a deep thinker and prolific writer. He had excellent communication skills. More importantly, as importantly, he promoted the careers of young scientists. Oh, we lost him. Yeah. Okay. His personal philosophy was in research. He only did research that no one else was doing, and only research that benefited mankind. He established the Barrows Research Institute in 1982, retired from MSU in 1997. He was author or co-author of 155 publications, 67 of which were in the platinum area. He was also inventor or co-inventor of 22 patents, and he was the recipient of 16 prestigious major awards. Although hopeful and recommended and certainly deserving, he was never awarded what many scientists thought was his inevitable and deserved prize, the Nobel Prize. Loretta Van Camp was a wonderful lady and a very talented co-worker of Dr. Rosenberg. She obtained her BS in medical technology from MSU in 1948, then took a medical technician position at the Michigan Department of Health. She joined MSU in 1960 as a faculty specialist in the biophysics department, working with Dr. Rosenberg as his laboratory manager. She was indeed very intelligent with particularly keen observation skills. She was, of course, a co-discoverer of cisplatin. She received in 1998 the MSU Distinguished Alumni Award. In 1999, she also was honored with the Honorary Doctor of Science. She was a philanthropist. She supported research in the biochemistry department for many years and supported scholarships at MSU. She was featured in what was known as Face to Face with Success, a television segment that focused on MSU alumni and their mentors. Cisplatin, which you will hear a lot about today, is the drug name for the square planar complex cis-diamine dichloroplatinatum 2. It's a square planar compound whose structure is shown here in the upper left-hand corner of the slide. It was first prepared by Pyrone in 1845. Thus, when you hear reference to the discovery of cisplatin at this symposium, what it actually means and is referring to is this discovery of the biological activity of cisplatin. The discovery of cisplatin, indeed, as you have already heard, was an important historic discovery that's had an enormously beneficial impact on the lives of countless cancer patients, particularly with solid tumors. We'll hear a lot about the benefits of cisplatin and carboplatin in the upcoming talks this morning. The discovery was made in the laboratory of Dr. Barnett Rosenberg in the biophysics department at MSU in the early 1960s while studying the effects of an electric field on dividing E. coli bacteria. The discovery team included Dr. Rosenberg, Loretta Van Camp, Tom Krieges, Andrew Thompson, and Eugene Grimley, all three of whom are here in the audience today. It was an accidental or serendipitous discovery, as you see was frequent in this study, and occurred because of three things: keen observation skills, particularly on the part of Loretta Van Camp, a prepared mind of Dr. Rosenberg, who was open to the unexpected result and intuitively knew what that unexpected result meant, and then led a disciplined and relentless pursuit of the cause of this result. This search indeed is a textbook example of the practice of the scientific method, and we shall move on. In 1960, while still at NYU, Dr. Rosenberg conceived of the idea to study the effects of the electric field on mammalian cells. I said mammalian cells. The driving force was twofold. Dr. Rosenberg was fascinated with the similarity of the mitotic images shown here on the left of mammalian cells and the lines of force and magnetic field shown on the right. He wanted to address a speculative notion held at the time that a magnetic component might be operating in mitosis in mammalian cells. So he decided to test whether the magnetic component of an electric field could interact with this suspected magnetic component in cells and affect cell division. The start of the discovery adventure took place in the biology research building in the 1960s, located near the chemistry building, which yesterday was right near where we had the poster session. These are photos of the original continuous culture apparatus used to study the effects of an electric field on E. coli bacteria in the basement of the biology research building. With the help of Professor James Trosco, here today, then a graduate student at MSU, he helped Dr. Rosenberg obtain all the equipment that was needed to carry out these early bacterial studies. The apparatus, complete apparatus, is shown here and consists of an audio oscillator and generator. This screen is very, very sensitive, I'm sorry. And a dual trace oscilloscope, and the actual continuous flow cell mounted in a water bath held at 37 degrees. Close-up of the actual cell itself is on the right-hand panel, as you can see. The next slide shows you a schematic that gives you a much better idea of the basic design of this continuous flow cell. It's a chamber made of glass. It has an entrance and an exit for the incoming medium C media used in the system. An inlet tube allowed air to come in and mix the solution, provide oxygen for the bacteria. It had 2 platinum electrodes dipped into this solution, and a voltage, AC voltage of 1,000 cycles per second and 2 amps were impressed across the electrodes to generate the field. Dr. Rosenberg was fully aware of the possibility that electrolysis could occur. Thus, specifically, he chose platinum as the electrode material because of its chemical inertness, and he selected the AC frequency in order to eliminate both electrolysis and electrode polarization effects. Experimental work began in September of 1961. In an early experiment, the most significant observation was made. When the electric field was generated in the bacterial chamber, the population density of the normal bacteria, shown on the left, began to decrease after 1 hour, and then the field was turned off. Microscopic examination of the effluent from the cell revealed that the cell division of bacteria was inhibited, forcing the growth of bacteria into long filamentous forms up to 300 times their normal length. This discovery then triggered an all-out search for the causative agent. But what was not serendipitous to Dr. Rosenberg was the potential discovery that the agent causing this effect might have utility in treating cancer. Unquestionably, the discovery and development of cisplatin is a monumental achievement, indeed the most outstanding discovery in the history of research at MSU. This discovery is a fascinating 3-part story consisting of the accidental discovery, the cell division in E. coli bacteria was inhibited but not cell growth, the search for the causative agent, and, in my estimation, the fortuitous discovery of cisplatin. Shown here are Dr. Rosenberg and Van Camp in the mouse room carrying out some experiments. The search for the causative agent was begun by Tom Krieges, who investigated all experimental factors that could potentially affect cell division. Factors previously known to induce filamentation were quickly determined to be not operative, such as osmotic pressure, effect of chemicals in the solution, and things of that nature. The electrical field itself, as important as it seemed to be very early on, was found to have no direct effect on the system. Thus, the causative agent was created by electrolysis. Oxygen was found to be essential for filamentous growth, not just for the livelihood of the bacteria, but for another important reason that signaled an oxidizing agent was in fact produced in the media by electrolysis. Tests for specific oxidizing agents that could have been derived from components in the media by electrolysis were found to be negative. Next, the role of the components of the medium C were evaluated, and it was found that the medium must contain the ions ammonium ion and chloride ion for filamentation to occur. The testing for the presence of platinum-2 and platinum-4 by Tom in the media exposed to the electric field led to the conclusion that there was a high probability that platinum-4 was produced in the ammonium chloride solution. Then a hypothesis was advanced that the platinum electrodes were undergoing electrolysis and that ammonium hexachloroplatinate, shown in red, was proposed as the causative agent. Ammonium hexachloroplatinate indeed tested positive as an oxidizing agent using the starch iodide test and caused elongation at a concentration of 10 parts per million over a period of 2 hours. Studies of the direct effect of ammonium hexachloroplatinate then began immediately. A paradox soon arose. The steady state concentration required to inhibit cell division and induce filamentous growth in the bacteria under the influence of the field was found to be about 8 parts per million. However, the test tube experiments performed using stock solution of ammonium hexachloroplatinate only induced inhibition, cell division, and filamentation at or above 10 parts per million. The paradox was never resolved at this stage. Shortly thereafter, however, 2 key observations were made by Loretta Van Camp. The characteristically pale yellow stock solutions of ammonium hexachloroplatinate had turned almost colorless and became more effective at lower concentrations of the stock solution. The increase in effectiveness also occurred only when the solution of ammonium hexachloroplatinate were exposed to sunlight. Hence, the stock solutions of ammonium hexachloroplatinate, in retrospect, had been stored on an open lab bench, exposed to sunlight over a weekend. The implication was clear. A photochemical reaction had occurred, and the photo products were more effective than ammonium hexachloroplatinate in inhibiting cell division. Eugene Grimley then investigated this photochemical change. He irradiated a solution of ammonium hexachloroplatinate at 5 parts per million with UV light at 365 nanometers as a function of time. The changes in the absorption spectrum clearly showed that a photolytic reaction had taken place. These are the absorption frequencies of the starting material. And as a function of time, they disappeared. The starting material completely disappeared with the formation of intermediate and finally a photostable compound in blue, whose absorption spectrum is in blue. The photo products from the irradiated solutions were then separated by what was known as high voltage paper ionophoresis, like electrophoresis, and visualized with stannous chloride. 3 species were identified. The starting hexachloroplatinate ion, which immediately changed to form a negatively charged intermediate and also a neutral species, the final photostable species. After only 1 minute of irradiation, 2 species, in addition to the starting material, had formed. Within 5 minutes, the hexachloroplatinate ion was completely converted to the intermediate, and then after 45 minutes, the only substance remained was the neutral photostable species. Then, using the technique of preparative ionophoresis, the same 3 products were separated, collected, spectra recorded, and tested in bacterial cultures. Again, these species were the starting material, the hexachloroplatinate ion, the singly charged negative species, and the final neutral species. The photoproducts could be accounted for in a 2-step reaction shown here, in which ammonia replaced sequentially 1 chloride to generate the intermediate, negatively charged, singly charged intermediate, and the final photostable product. Bacterial tests were then carried out to see what their effect was in terms of inhibiting cell division. Indeed, the hexachloroplatinate ion was not found to be an inhibitor of cell division, but rather a bactericidal agent. The monoamine pentachloropplatinum single minus ion was found to be neither a growth nor cell inhibitor, and the neutral diamine cis-diamine tetrachloroplatinum is a potent inhibitor of cell division. A manuscript was then submitted for publication, and at this point was outrightly rejected. The editor's explanation was the formulation of the species that was a potent inhibitor of cell division could be in form of two isomers with the same formula. It's both the cis and trans form of the diamine tetrachloroplatinum 4, and that the authors were instructed to find out which of the two isomers was the active isomer. So synthesis began. Andrew Thompson accepted the challenge and prepared both platinum 4 isomers by first preparing the precursors, the trans and cis dichlorodiamine platinum 2. He oxidized them with chlorine, and the compounds were then tested against bacteria. And indeed, both trans isomers, the trans platinum 4 isomer, was found to be ineffective in inhibiting cell division. The trans dichlorodiamine isomer was found to had no visual effect on the E. coli bacteria. But when the testing of cisplatin compounds occurred, then the platinum 4 analog, the cis-diamine tetrachloroplatinum 4 here, indeed was active. It was the active neutral entity. In testing the precursor to the platinum 4 compound, however, it was found to be a very effective inhibitor, a more potent inhibitor of cell division than was the platinum 4. This indeed was the start of the realization of cisplatin as a potent anti-tumor drug. Cisplatin was then tested. Rosenberg extrapolated from bacteria to mammalian cells and suggested that cisplatin be tested in mice to see if it could inhibit the division of cancer cells. Now, these first tests were carried out in mice using sarcoma 180 solid, actually in ICR mice, and then subsequently at the NCI. The testing of the platinum 4 compound showed activity at 10 milligrams per kilogram as a daily dose with a T/C of 29. But the cisplatin showed a much more potent dose, much more effectiveness, being effective at a dose of 2 milligrams per kilogram, daily dose, giving rise to a T/C of 1.8. The T/C is the ratio of the weight of the tumor of the treated group to the control group, and therefore the lower the value, the more effective is the compound. Finally, the suggestion was made. Loretta carried out an extensive set of tests in which she now tested cisplatin using a much more stringent set of conditions. She first implanted a tumor, 50 milligram tumor in the control mice in the top panel here. Going to completion, by day 21, all these animals died. The bottom panel shows a progressive and successful regression of the large tumors, which treated on day 8. The tumor already had a lesion by 12, was groomed out or resorbed by 16, and by day 36, the mouse was totally cured. The impact and discovery of this development generated enormous interest, both research in metal-based drugs, in university, government, and commercial entities. It opened up sources of grant support for students for their study. It brought about a renaissance in bioinorganic chemistry, to be sure. It stimulated interest in cancer drugs in the pharmaceutical industry in general. And it overcame the stigma that all metal-based compounds are heavy metal poisons and devoid of biological activity. And most importantly, it provided in cisplatin a needed and effective drug that has benefited millions of patients afflicted with solid tumors. Thank you. I'll quit. Thank you for your time and your attention. I could add one more comment. I've got just one or few more minutes. This is an important comment. This is a comment made by Dr. Bruce Chabner, who was a former NCI director. His comment was, it would be frightening to imagine the current status of cancer treatment without this essential drug. And of course, this could apply also to carboplatin, a drug that follows cisplatin, is also very, very effective. Pardon? Oh, yeah, I'm sorry. Something happened to the... There we go. Sorry about that noise.
Oh, thank you so much. Do we have any questions right now?
Outstanding, and what a wonderful example of team science way back in the 1960s. In the 1960s, there were many wonderful experiments in vitro and in animal models of promising agents that turned out to be ineffective in human situations. I wonder if Barney and his team ever had any idea before the first human trials what a profound effect this would have upon human cancer.
No, I can't answer that question directly, but there was a hope and dream and actuality of that happening. I'm sure Dr. Rosenberg and his team had great hopes that the cisplatin would be an effective and useful and approved drug. I can't speak beyond that. I never really asked that specific question to Dr. Rosenberg, but I'm sure the answer is yes.
Can I say something kind of related here? Back when you were when folks were in the lab trying to figure out what was the structure, what's the molecule that's leading to all these great effects, what were you guys thinking was the mechanism? Why this molecule was having that effect? How it could be having that effect?
That's a great question, because we just happen to have here in the audience Harold Harder, whose work was devoted to the beginning of the examination of just what the target site was and some insight into the mechanism of action. His work identified DNA very early on that in the fact that cisplatin inhibited the incorporation of bases into the DNA template. So that was the start of a small opening of a window to see what the mechanism was like. And as we're here today, Steve Lippard took up that task years after that and pursued it with diligence and depth. So
you should have zoomed in on the DNA very rapidly.
Pardon?
They zoomed in on the DNA very rapidly
Yes, he did.
in the lab.
So Jim, Rob, Jim, so you didn't talk about carboplatin at all, but earlier this morning, Jim Trosco and I were discussing the serendipity that led to cisplatin. What about carboplatin? Was that rationally designed, or briefly, how did that come about?
Sorry. Carboplatin was made in 1971. Mike Clear and I were doing a structure-activity relationship. Mike actually made carboplatin. It lay dormant for many, many years because it wasn't dose potent. It was 10 times less potent than cisplatin. And at the time, we were trying to find drugs that were equally active as cisplatin, but also dose potent. So the dose, so it was put on the back shelf for a while, until probably in the late 70s when it was realized that there needed to be a second platinum drug to replace or augment cisplatin because of its toxicity. And so that's when Mike Clear and the British group took up the mantle of looking at and identifying what might be a second generation drug. And so 50 compounds were looked at first, got whittled down to 4, and out of that 4, based on activity and toxicity, carboplatin emerged.
My name's Andrew Thompson. I just want to say a little bit more about the uptake of cisplatin by the clinical community. In fact, it was rather slow going at first, and certainly the NCI were extremely reluctant at first because of this supposed heavy metal poisoning. We'll hear, of course, from Professor Einhorn later on what did trigger it. But before that, the Chester Beatty became very interested in cisplatin through its then research director, Sir Alexander Haddow. And Haddow had been working with classical alkylating agents. But he had an interesting paradox that says that those compounds that cure cancer can also cause cancer. And Haddow had consulted with the Mond Company in Wales, where nickel carcinogenesis amongst the workers was found. And Haddow saw the Nature paper from Barney, and he knew that platinum and nickel were in the same periodic group, which is rather good for an experimental clinician. But he made that observation, and he wondered whether this was perhaps another example of Haddow's paradox, that nickel caused cancer and platinum could cure it. And so he took up this compound, and the Chester Beatty, I think, may have been the first to test it in humans. Certainly, it was tested against ovarian tumors in women patients. But of course, it was the work, as we shall hear from Einhorn, that really set the world alight, as it were.
That was Andy Thompson, who was part of the discovery team. Just to elaborate just a little bit, the resistance on the part of the NCI was so strong to test cisplatin. They thought of cisplatin, as you put it, a heavy metal poison. And they waited until the Rosenberg results came out and the Chester Beatty results came out before they actually began any kind of testing and evaluation of cisplatin.
So, quick question, did the lab ever go back to studying the effects of electric and magnetic fields on cells? Or was that was the end of that biophysics thread?
It's funny you should ask that. I was talking with Matt Hall just yesterday. We've been thinking about reinvestigating that whole system. I have not found a collaborator at EMU, a biological collaborator, who'd be willing to do this. So we may look into that. I would really like to know exactly what is in that solution as a function of time all the way through the process of the inhibition and then formation and filamentation in terms of the chemical species. Because there were a lot of false publications saying that cisplatin was produced in that media, and there's no basis for that statement whatsoever. And knowing the chemistry, as so many people here know, it's unlikely that cisplatin would have been produced in conditions where the amperage was 2 amps and the voltage was very high. So that's just my own comment.
One more question?
I'm Tom Krigas, and I can recall the day I walked in the lab, and Barney and Loretta were talking about making the connection that this is very likely a possible anti-tumor drug. And of course, I was the smart ass, and I said, "Oh, you are really stretching this." Well, you know who was right. But I recall also that money was always an issue in conducting the research. And Barney did 2 things that were unique and caused some problems, which he often did. He went directly to the drug companies, 2 major drug companies at the time working with platinum, and got them to help fund the research. And he still needed additional cash, so he went to a major network station, and they broadcast a report of the research at that time. And that evening, he and his wife, Rita, had a small gathering at the house, and we were just talking about how that had gone, and he began to receive phone calls from around the country. people had seen it. And they wanted their hands on that drug. And it was 15 years away from being commercially available. So not only did Barney and Loretta think that this was going to be an anti-cancer, the general public seized on it too. So it was only NIH, I guess it was, the National Cancer Institute was against it. So that was it, just a couple of reminiscences.
Thank you. Let's thank Jim again.